Good morning, good afternoon, everyone. Marina Kirkitadze is here from Sanofi Pasteur, Analytical Sciences Department, and I represent Analytical Process Support and PAT platform that was formed almost a year ago. And right from the beginning, we got the opportunity to work on the PAT tools in collaboration with our colleagues from manufacturing technology and from METTLER TOLEDO, who, with great help to bring us up to speed and opportunity to learn a lot on our product, what we can do and how helpful we could be for the processes. So on the next slide, we could try to convey the purpose of PAT. So PAT stands for Process Analytical Technology. Why do we want to use it is to support full cycle of vaccine manufacturing, starting from raw material to media, bulk, active ingredient, formulation, filling, packaging, and release. The estimate that we have in the company, and I believe it's also the benchmark for industry, that approximately 10% of vaccine production time is dedicated to testing and process monitoring. And really, if you think about it, it's quite a lot of time compared to, just 30% of the time spent for production. So obviously, by introducing PAT tools at different spots, or if we could cover everything by PAT tools, we could significantly decrease the production time and become as fast as our processes are. And so obviously the ultimate goal is to develop and implement online and inline technologies to measure critical material attributes and critical quality attributes in real time. Obviously, ambitious goal is to have real-time testing, real-time product release, but we're starting with something that we can achieve and can try first is critical material attributes and in process monitoring. Once the knowledge is built, we can become more solid in our move of the next step, which is real-time release. So with that, first, let's look at the examples of current offline methods. You can't really perform any tests inline unless you have a very good understanding what offline tools usually available as lab-based instrumentation, quite sophisticated tools, but very often with a simple output, very easy to use. Of course, training is required, but all the mathematics often hidden, and years of development and evolution is again reflected in the software, which is quite comfortable, can be used by the operators to generate data. So the tools, for example, like Raman and FTIR, can be used for raw material identity, fermentation media. We see mostly use near-infrared, but we can also use Raman. And YSABRD is Bioprocess Research R&D, which we have here with Formulation Stability Group, kindly offers to have access to the NIR instrument offline, so we can gain understanding, but this technology is applicable. The adjuvant structure, antigen structure, we can assess secondary structure by infrared. If we go into adjuvant and adsorbed drug substances, which is adsorbed proteins, infrared is helpful again because it provides both secondary structure analysis, but also presence of adjuvant or any excipients that you add during the process, and those are reflected in the FTIR spectrum. Nano-DSF, again, used both for in solution proteins and also for proteins in adsorbed form, because a nano-DSF with a fluorescence readout can quickly produce the answer and both report tertiary structure and also conformation, and also the thermal stability. DLS is more for sizing of the protein. In some cases, quite important. So DLS we use as a fast technique, but if you need to go deeper, of course, there are size exclusion with the mouse detector available. But in terms of applicability, DLS is considered because it can be used as online tool. Now DSC is more for offline. With automation can be used with robots, but nevertheless at present state DSC obviously is remains of us offline lab-based technique, but it gives good insight into tertiary structure. Laser diffraction and phase analysis, light scattering or pulse come handy when you work with adjuvants, adsorbed form, and Raman as well. Raman spectroscopy can be very helpful when you have adsorbed proteins. The only difference is that all these preceding methods you work with suspensions as these, whereas for Raman you need to dry this the suspension, so you only work with in dry form or solid form. So the purpose is to build the foundation of inline probes, or for online solutions. And the reason for conducting small-scale studies and offline studies for the materials produced at manufacturing scale, or at scale as we usually refer, these type of studies are not permitted in production facilities. So you really need to have a lab space and instrumentation, people, trained personnel, and also the I think the energy in this team to very quickly assess what technology can be done, can be used for specific case. And so with that, offline techniques basically your foundation that you have to build first to speed up the development for the inline solutions. So therefore inline solutions as well, basically they stemming from their offline tests. They like engineering wise they designed in more how to say miniaturized or adaptable form, but the results they produce are comparable to what we have in lab-based methods or offline methods, and that's really promising from what we had so far with near-infrared, with infrared, and with FBRM. That infrared, mid-infrared, and FBRM would be discussed in this topic, in this presentation. So the purpose for evaluation of the inline probes is obviously a real-time release test. As I said, it's the most ambitious goal at this point. For automation and control, for continuous processing, and for consistency, lot-to-lot consistency or batch consistency. But reality is that first of all, inline tools give opportunity to look inside of material making during the reaction and at the final stages, and also to build the large data sets that could be used for trending, for basically predicting how process could be given initial parameters. So that's really the and as I said, the ultimate goal is to gain understanding and confidence that some of the techniques that are used for inline monitoring at the certain points where they report not just critical material attributes but also critical quality attributes, they can be evolved into the release tests, inline release tests or online release tests. So the outline for presentation is characterization of aluminum phosphate adjuvant. Outline was done by particle sizing by laser diffraction, inline by focused beam reflectance measurement, particle track probe, outline composition by Raman and FTIR, and inline by infrared probe. Characterization of adsorbed protein is particle size by laser diffraction and composition by Raman and FTIR, and characterization of protein adsorption by particle track probe FBRM and by infrared inline probe. The purpose of the study obviously to gain knowledge about adjuvant surfaces and antigen protein distribution, basically by adsorption. Why we're so interested in it is it's linked to antigen adsorption capacity and possibly immunogenicity. And so to examine the effects of adjuvant protein antigen structure, outline and inline is to assist in determining product expiry date. So with aluminum adjuvants, why we're interested in them? Because they're used in vaccine formulation over 60 years. So it's really the compounds that are long-term available, used by many manufacturers of vaccines. However, like over time, they started, I think, with aluminum sulfate, which was more popular early on in 1926-27 when they just started using it. And now it's more aluminum phosphate and aluminum hydroxide, although aluminum sulfate in certain formulations you see, but more like as a mix with aluminum phosphate. So why is it used? Because it introduces early, high titer and long-lasting immunity, and most widely used as adjuvants in veterinary and human vaccines. During all this time, when aluminum phosphate, aluminum hydroxide adjuvants are used, there's billions of vaccine doses were administered over years. They are regarded as safe when used in accordance with current vaccine schedules. And references here to both WHO, World Health Organization, and also to their review by Elderman and reviews infectious diseases. So references are here. So just to start, as discussed on one of these slides just before, we started with laser diffraction as offline tool. We do it now probably 6 years. Over last six years, we gained good understanding of adjuvants profile, size distribution, basically several lots, with aluminum phosphate close to 50 lots, if not more at this point. So it's basically quite solid understanding what technique can do for you and what you can expect. And so with that, what you see here is we have on the left hand side panel is aluminum phosphate intermediate, which is basically right after mixing of aluminum phosphate, sodium phosphate and aluminum chloride raw materials to form the adjuvant. And then after several steps, its final aluminum phosphate adjuvant is produced, and basically size distribution measured for that again. And that's the compound that is used after for adsorption of the proteins. And so literally there are no significant changes in particle size before and after processing. The shoulder that you can see is slightly changing. It doesn't really, shift. It's in the same position, just more prominent at the intermediate stage and more subdued. But in terms of DV50 and DV10 and DV90, the criteria that is used for analysis with mere equation is not really changing. So therefore, conclusion is that during production, once the aluminum phosphates formed, the size is same throughout the entire reaction. And then here is basically introduction to focused beam reflectance measurement probe or particle track probe, and that is basically a probe containing a laser source, a fiber, detection fiber, beam splitter. It has a tube, a sub-5 window, and that produces focused beam. Rotating optics produces focused beam, and this focused beam basically shines laser light into the solution, which is gives you kind of like a circle or circular snapshot of in situ distribution of particles in given substance. In our case, obviously, it's aluminum phosphate. What you see here is the image with a particle view probe, just to illustrate what FBRM would see. So it will see particles, so you could measure the size of them by measuring the Hort, and also you can count particles. So FBRM can work like laser diffraction and Coulter counter in one. So size distribution and particle count. So laser diffraction and FBRM analysis of intermediate and final aluminum phosphate. So on left hand side panel, you see the FBRM and laser diffraction in blue and orange colors respectively for the so-called small scale aluminum phosphate. And we know when we produce it small scale, it's usually larger size, between 27 almost 32 33 microns, whereas when they produce that scale or manufacturing scale, which is more than 200 liter scale, it's basically quite consistent, both by laser diffraction and FBRM, and the size by laser diffraction is 10 to 12 micron, and then for BRM we see 17 to 20 micron. So there is no significant difference by each method between the particle sizing before and after processing. However, there is a small difference between the two techniques, and one probably would expect it, especially considering that for laser diffraction you have to dilute the sample significantly, almost 100-fold, in the dispenser before you do the measurement. But when you do the measurement with FBRM probe, you measure in situ, so as is, material as is, so therefore you measure neat sample, so the size obviously slightly bigger. So that's our explanation to differences, and most likely that is also advantage that when you analyze in situ, you really can see the aluminum phosphate in making and measure its real size. Diffraction results, however, are important because they reflect the size that in more diluted way. So when you mix it with adjuvant, with the protein, and becomes more diluted, that is just as relevant for test. So here we have FTIR analysis of aluminum phosphate adjuvant manufacturing stages. See on the left-hand side is the overall spectrum, and you see this almost no change. But when you zoom in an area between 1200 to 800 inverse centimeters, and look at the peak around 10, 69, you see that it's moving. So for the intermediate, we see the peak at 1067, and then for the final it's 1076. So about 9 centimeters, inverse centimeters shift to the greater wave number indicates that PO bond is strong after processing. So that is done again often. So now what we can do inline. So again, here is a React IR, it's an introductory slide, just to see, okay, here is equipment. It's obviously miniaturized compared to benchtop instrument that would use, and also the ability to penetrate directly in situ and measure samples, materials as is, as they made is offers great advantage. So here we have 7 reflections, whereas the results shown before, they measured by a crystal with 3 reflections. So 7 reflections are important because they give more sensitivity, and considering that inline you would measure the samples that are moving, because they are mixed, continuously mixed, and they don't really sediment on the probe, or they stay too long near the probe. That means that the result, the signal will be weaker. So 7 reflections can intensify the signal and basically gain better result. So here is the snapshot how aluminum phosphate experiments were done. So here is EasyMax, the small reactor produced by METTLER TOLEDO, and so you have 2 chambers that you can operate. You have a mixing pump on the right hand side. You can see it, the white box with black dots and with a green beam kind of in the middle, and so it's connected to the titration pump, or the syringe pump, if you will. And so we have the samples of aluminum chloride and sodium phosphate introduced with certain rate, and so we can optimize or see how the reaction changes at different conditions. So for the to measure the reaction and the final aluminum phosphate characteristics, we used initial 2 probes, both Raman and FTIR. We obviously had higher hopes for FTIR because Raman, we know, at least offline, we always work with solid samples. So we precipitate the suspension and dry it, and then analyze it as dry. But then here we decided that maybe once it's in situ we may have better chance, so why not to double check with Raman as well. So all the processes were captured into ICE control and available in ICR and IC Raman. And so infrared spectrum, I have to say up front that although we collected Raman for raw materials, for reaction it was not sensitive enough as we expected, and so we basically proceeded all the other experiments. You will see they're done by infrared. So infrared spectra for aluminum phosphate reaction references. So we have peak for sodium phosphate, which is at PO peak is 1004 inverse centimeters. For aluminum chloride is 960 inverse centimeters. So I'm just stretching attention on the sodium phosphate peak because that's what we'll monitor further in the coming slides. What was interesting for us is because here you can start with soluble sodium phosphate and monitor how it converts into the PO part of it, at least converts into the solid phosphate, and how peak changes over time. And so that was also the mark for us to monitor reaction from liquid to solid, and then after the treatment it was is subjected to the peak changes again to the greater wave number. And so here is a precipitation reaction with reaction trends. So you see that amount of aluminum chloride decreases with time, and it's shown by purple curve, and then the sigmoidal curve, and then blue sigmoidal curve is basically creation of aluminum phosphate, which is on the rise, and it tends to plateau at some point. So mid-infrared, we learned that can be used for monitoring the reaction from start to end point, and it's it can be also used to basically visualize whether all the sodium phosphate was consumed, or and if there is a retentate, like basically how much of soluble phosphate you still see after all the reaction is completed. So basically that's another opportunity to optimize aluminum phosphate or using these tools, which normally if you just do offline you may not even see, because you really need to see reaction. And so that offered us a window basically to see it and to monitor it and learn more. So then the next step was processing condition that impacts aluminum phosphate spectra. So we go back to the slide that you probably recall from offline studies, where we're zooming into the area between 12 to inverse centimeters, 12,000 1,200 inverse centimeters to 800 inverse centimeters, and focusing on the phosphate peak. So here, just to illustrate that after the treatment at a small scale study, it's a left hand side panel, you see the change, very subtle but nevertheless. And then on the largest scale, we see on the right hand side panel, we see that before and after treatment, it's basically there's a disappearance of the peaks at like a plateau, I would say. So plateau becomes more like a shoulder, and gearing towards the higher value or higher wave number. So obviously the change in mid-infrared inline is smaller than what we see by offline studies, but nevertheless it's consistent, and it can be used as a monitoring tool for the basically processing of aluminum phosphate. So for reaction first, which was new to us, and then also for processing, that we can relate to offline tool. So on this slide, we took a step further, and we decided to see the adsorption reaction that happens between the protein and aluminum phosphate adjuvant. From scanning electron microscopy, we know that Tetanus Toxoid is normally situated at the surface of aluminum phosphate adjuvant, and basically we were interested how the reaction happens, and what kind of changes we possibly can detect by infrared that we don't see by offline tool. And again, Raman was also used here, but as mentioned before, the results were not sensitive enough. I mean, the spectra were not resolving enough so that we could comment about changes, whereas infrared showed the differences in the MIT1 and the MIT2 regions, so that we can monitor and comment about secondary structure changes in the protein. So with that, the tetanus toxoid sample we got in large quantity compared to what we normally test in the lab-based instruments, and the aluminum phosphate was in the quantity scaled down from what normally manufacturing batch is, and that was done with help of Daniel Chapman and Zhemek Koval, who quickly recalculated the values for us so that we could proceed quickly. And so the reaction was set, and phosphate was added into the tetanus toxoid solution, and the reaction was monitored over 16 hours. So as a result, here is the adsorption of tetanus toxoid onto aluminum phosphate adjuvant, and it illustrates a screen capture from the ICE infrared React IR software that shows raw data. So it's you shows the trending over time, and basically it's on the left hand side panel, upper one. On the lower one you see the overall spectrum with a pink box illustrating the region where most changes are detected. Then on the right hand side you see the screenshot of individual graphs or individual files collected over the 16 hours, and then at the lower panel from the right hand side you see 3D illustration of the changes in the MI2 region that was detected over this 16 hours time. And if you zoom into that, maybe it's hard to see, but really what I want to stress is the MI2 peak. You could see that gradually changes over time, and it becomes smaller and smaller and more looking like a shoulder for the adsorbed tetanus toxoid. So tetanus toxoid adsorption spectra with subtracted water looks like presented here in this slide, and in the dashed line you see the reference spectrum of the tetanus toxoid, and that is from offline studies that is only used as a reference just to illustrate that both amide 1 and 2 peaks are present, whereas then you see it in line. It starts to decrease gradually, starting from pale blue, which is where the arrow is, into the gray. It's where arrow is pointing, and completely disappearing, more looking like a noise or shoulder in the peaks of that of green and gray color, and ending up with light like a prominent blue color, where you see just amide 1 region present and no amide 2. So basically the conclusion is that amide 1 and amide 2 peaks illustrate secondary structure changes, and amide 2 peak completely disappeared, basically significant decrease. And that goes hand in hand with our previous results obtained on offline study, basically by lab-based instrument, infrared spectrometer. The tetanus toxoid in solution has both peaks, amide 1 and amide 2, and whereas tetanus toxoid with aluminum phosphate illustrates just one amide 1 region and a shoulder from amide 2, and also presence of aluminum phosphate peak. And again we see increase in alpha helix and beta sheet, which is consistent with changing or basically tetanus toxoid landing on the surface of aluminum phosphate adjuvant. And so in conclusion, I think the key essence is that we can measure particle sizing by FBRM, and we see that it's a variable to be considered, meaning that in situ and data by FBRM versus lab-based instrument by less diffraction, we see difference. Nevertheless, difference is the results are consistent for the lots by each technique, and the difference, if you really think about it, is really small. However, it's there, and it signifies that when you measure something in situ, you have basically more closer to real material attribute, whereas when you dilute, perhaps you can test to diluted samples that potentially the size that is when vaccine is administered. So both techniques have value, but I mean for process specifically for timing and decision making about particle sizing, obviously FBRM has advantage, and that's something that we want to proceed further. Aluminum adjuvant functional groups are well suited for infrared analysis. As you can see from the slides, you can start from raw materials, sodium phosphate, and carry through monitor the peak for that and carry through into the reaction and formation of suspension, solid PO4 if you will, and basically changes further changes to that peak that undergo after the several treatment steps that occurs during the process. And so in addition, we found that FTIR can also report changes during adsorption reaction of the protein to adjuvant, and that offers the opportunity for greater process understanding and endpoint determination for optimization of the process in the long term. And so here is graphical summary of the process characterization of offline tools. So we tried in a pictogram fashion to illustrate the steps. Really you see the person appearing only at the lab kind of pictogram for the lab analysis, although as you can imagine that persons in human involvement is throughout the process, and we see at least 4 or 5 people involved in the process, just from bringing the sample, aliquoting it, bringing it to the test lab, testing it, analyzing data, reviewing, approving the report, and basically make it available for data file. And data file is a really generic term for anything that you can call from batch production record to certificate analysis or to characterization report, whatever you need to support data for release or data for license application, for example. So really, between the initiation and the endpoint, it's quite lengthy procedure, and that's why you see the small clock, a pictogram present everywhere, so it's a not instantaneous process. Whereas for process characterization with inline PAT, it's really instantaneous. You have material, and it's just a matter of the time that you need for the process, whereas results happen as you progress. So it's real-time data collection, gives significant advantage in terms of reducing cycle time, basically reducing the number of perhaps redundant tests, and also reducing time to decision making. And also for making it available for data file, it's really much faster option. Obviously the infrastructure has to be built, how it will be presented, what database will be collected, but the reality is you have to generate the data first and start to do trending, and basically in parallel work on how to capture data for long term, and also for processing further for any specific reports. It still gains you a significant amount of time, but more importantly, it gives you the knowledge about the process to the point that it's predictable, it's understandable, you can optimize it, you basically become more in control and more flexible in terms of moving the process or adopting it to different scales if you need, or moving it between sites. So that's a graphical summary of the you can find also in our paper. I also want to acknowledge Carmen May. I did it in previous slides as well, that she was really the key person to make it happen, and to put all the data together, to write a manuscript, to really work on the wording and make it, ensuring it flows. And obviously all this collaboration will not happen without presence of our manufacturing technology colleagues, Daniel Chapman, Nicole Lazaris, and Liliana Sampoliano, who provided the samples. Daniel was basically designing the experiments, how he wanted to do with a small scale or at scale to be tested, and Nicole helped with testing as well as being with us, so we do it everything properly and as per procedure, so it's completely aligned. And then Uli Richard and Sushin Kong for the particle sizing, and I have to highlight Sushin. He really did a superb job in terms of generating data, explaining to us, and demonstrating the value of FBRM probe. And then offline data by Moria Moore and Sylvie Moran from York University help to understand where on the surface with the scanning electron microscopy the samples would be situated, so where protein distribution happens and how it's on the surface. And so that helped to basically solidify the conclusion for FTIR, for example, offline FTIR that we had, so to basically confirm our results. And then Bruce Karpik, basically like a sponsor, a champion from Vita site, which is vaccine acceleration, innovation technology acceleration program that he runs the analytical domain, and PhD is part of this domain. So it was great help to see the context where we can contribute, not just here at our site, but also globally. And Matt Balmer, who's the analytical sciences head here in Toronto and for Swiftwater, also gave a vision that you can go beyond. You can look at what a different site is important, and so basically use the same information, same type of technology for different projects and different aspects. So that was really helpful to get the context, to get relation towards global organization that Sanofi Pasteur and Sanofi is. And so with that, we're really happy that paper did happen. It went through the scrutiny of peer review and it was accepted, and it's available online since 21st of August, so you can fetch it using the link below. And with that, I would like to move to the acknowledgement slides. Basically, to reiterate, in Sanofi Pasteur team you see more people than were listed in the paper, and that's basically their contribution was not throughout the entire experimental setting that we had, but nevertheless it was important. Like Zhemek helped with quick formulation of tetanus with aluminum phosphate, scale down, so it's achievable, and Jessica a lot of the logistics and testing offline, so that would gain us confidence. And of course, I think it's the first time we have it, but I also have to say that without METTLER team who basically very quickly presented all these wonderful techniques and easy marks to do the reaction in line, nothing of that would be happening. And I have to highlight the contribution of Katerina Droleviva. First of all, she really brought the team together. So she first brought Sushin, who helped with FBRM, taught us, and then Jim Crow and Will Kowalczyk and Stefan Woods was the second team who came, and it was a really vibrant group of scientists who really stimulate and motivate you to work. But also I think with the context of PAT, it's worth mentioning that PAT is not just instrumentation and inline probes, but the fact that you have people in the room who are professionals in their own specific subject, but they can share the knowledge and bring you up to speed in a very quick manner. It's a human PAT that also happened during this study, and I have to acknowledge that. So thank you very much for your time, and I would be really happy to answer questions and participate in discussion, and look forward for the fruitful discussion. Thank you.